Dear students, welcome to. Today we're going to start studying the DNA, but not in the all types of the cells in our body, but just in the somatic cells. And also we can get the difference between some types of mutations within that happens and occur for the human and also the plant. Let's start the lesson today. The human somatic cells contains about 46 chromosomes, okay? And right now we need to know how these groups of chromosomes, histones and non-histones, they are packed and they are intensed and condensed inside the nucleus. If we imagine that the DNA double helix from these chromosomes where they are aligned or became a linear, stretched out, it's gonna be about 2 meters in the length, okay? While the histones and other proteins has an important role in the backing, okay, the high length of this protein, okay, to package it in just a tiny diameter about from 2 to 3 micrometer. But biochemical analysis and also the electron macrophages, they are shown that the DNA is wound around a clusters of histones. The clusters mean what? Clusters mean a group of histones, and also we have two types, as we said, we have histones and non-histones type. Yet the forming a string of particles called nucleosomes. Again, we're going to know how the proteins or how the enzymes play the important role in the backing of this length inside just a tiny length of 2 to 3 micrometer. This shortens the molecule about tenfold, but it must be packed about 100,000 times more tightly to fit into the nucleus itself. So, the string of the nucleosomes is coiled so as to pack them nucleosomes together to form the nucleosomes. However, even this is not sufficient to shorten or to condense the DNA molecule to the length actually observed. The tightly coiled strings of nucleosomes are arranged in large loops, and there are some actions and some enzymes they are work on these large loop, okay, to intensify and condense the length of the DNA. Believe or not, that to be held in place by structural non-histones proteins of the chromatin. The chromatin packed up as tightly as possible is said to be condensed, highly intensified. When the DNA is in this state, in this case, the enzymes apparently cannot get at it. So, if you can imagine that a missed part or damaged part of your DNA, and you need to repair this part, this part of DNA, it will be unwounded, okay, and it lined or it became a straight line, because the enzymes, it cannot work by this intensifying or this kind of condensation. The package must be unwound at least into a string, okay, or as we said it, nucleosomes, before the DNA can serve as a template for making or manufacture DNA or RNA synthesis, as we're going to study in the next chapter. And we have here a picture to show what the kind of condensing DNA and formation of nucleosomes and formation of chromosomes. Okay, the upper side or the upper picture, it shows that these pictures are taken by I think definitely they are taken by X-rays. Okay, and the first one is the form of chromosomes. After that, after unwounded the chromosome, the intensifying of chromosomes to get the chromatin. Okay, and the third one is, or the fourth one is the condensed DNA, and finally we have here the histones. Okay, and this is the form of the DNA, or the double helix. Okay, what about the structure of the genome? We have some information about the human genome and how the human genome affects the everything in the human being, of the human life. Actually, an individual's genome consists of the total of all genes, okay, and also of the DNA in any one of its body cells. I mean that if you have one cell from any part of your body, I need you to be sure that this cell, it contains, for example, if you have one cell from your hair, okay, and the other cell from your small intestine, both of them contains the same amount and the same content of the genetic information or genetic material, okay. But here is it, we have something I need you to try to imagine it. The cell in your hair, it contains the all genes, okay, but most of them, they are blocking off, except the part or group of genes that's responsible for growing and appearing the features and characters of the hair. This on the same sequence also, the genes that the cell that has been taken from the small intestine also includes the same sequence and the same amount of genetic material. Okay, most of them, they are blocking off, except what your body needs in this part. In 1977, the researchers found a method of determining the sequence of nucleotides in DNA and RNA molecules. This provided the tools to describe how the genes are arranged within the cell's DNA molecules. We have already met some parts of the genome. Yes, in the last time, okay, we already have ability to isolate some of these genome. Many genes carry instructions for making proteins. Most of them, actually, they have information and data for making and manufacture of proteins. Others dictate the sequence of nucleotides in the ribosomal RNA, which enter the building unit or the building of ribosomes and transfer RNA, okay, which is carried the amino acid during the protein synthesis, as we go into study in the next chapter, how the proteins is synthesized inside your body. Actually, in prokaryotes, genes dictating RNA and proteins synthesis, they make up most of the genome in case of prokaryotes, but in case of eukaryotes, we have another sequence. However, in many eukaryotes, less than 70% of the genome, they are serves these functions, and the rest of the genome is unaccounted. The researchers also found that the identification and the given name for a various kinds of non-coding DNA. Non-coding, it's mean they didn't get a distinctive function for this part of DNA. So DNA that does encode for RNA or proteins, we call it non-coding DNA, but we still have much to learn about their functions. Okay, and actually, if you're going to search or browse on the internet, you're going to find a lot of searches that discuss this part of the genetic engineering field of non-coding DNA. Actually, we call them non-coding, but they have a function, but till now they are unknown yet. Well, we have here the repetitive DNA, okay? And how the DNA is repetitive, and what is the importance of this part of the DNA. Well, most genes are present in only one or a few copies in the genome, in the human genome, okay? But every eukaryotic cell carries many or hundreds of copies of the genes needed to synthesize ribosomal RNA and also histones, okay? These are molecules that the cell needs in large amounts. Again, each cell needs these molecules in large amounts. Well, and it is reasonable, I mean, it's logic to suppose that having a multiple copies of these genes speeds up increases it looks like, or acts as a catalyst, okay, to speed up, to increase the speed of the cell production of new ribosomes and also histones. This is one of the main functions of the repetitive. DNA also sequencing has turned up many more repeating sequences of DNA, and also the role of most of this repetitive DNA is still unclear, or as we said it before, it is still not unclear, but we didn't study the main function, the main role, okay, inside your body, okay? For instance, the fruit fly Drosophila, well, Drosophila, well, it's a type of fly. Most of the genetic experiments or genetic engineering experiments are carried out on this fly, which is called Drosophila. And this fly, mainly it lives on the fruit, okay? It feeds on the fruit. So they are called fruit fly Drosophila. The brief nucleotide sequence here for this fly, adenine, guanine, adenine, adenine, and guanine. This one is strand. So if DNA, you can conclude that the other complementary side, which is what? It complementary to the, yes, thymine and guanine by cytosine, thymine, and cytosine. It repeated about this sequence, okay, in the fruit fly Drosophila, repeats about what? 100,000 times in the middle of just one chromosomes. Okay, imagine the remainders, the rest of the chromosomes, how many times this repetition or repetitive DNA is repeated in the other chromosomes in just one living organism. These and many other repetition and repeated sequences are non-bonding in DNA. Other non-bonding DNA that we're going to study, the DNA also in eukaryotic genomes, they are sure they contain a great deal of the other non-bonding DNA. Even before the DNA sequencing become possible, I mean before the scientists believe that they have ability to study the DNA sequence, the arrangement of the nucleotides on the DNA information of genes. Well, the scientists of the genetic engineering observed that the amount of DNA in a species genome bears, or they carries, a little relationship to the complexity of the organism, of the number of proteins which is produced. And remarkably, a little of the DNA of the plants and animals, they are actually codes for these proteins. For example, the largest known genome belongs to what? The salamander. Okay, salamander, it's a kind of amphibians. Okay, its cells contains about 30 times the amount of DNA found in human cells. Okay, so this is the largest known genome right now. It belongs to what? The salamander, one of the amphibians. Okay, although they produce a fewer proteins, I mean, perhaps some of non-coding DNA act on keeping chromosomes structure as it is, or as a kind of stability of the DNA structure. Some regions or areas of DNA are references to places at which the mRNA or messenger RNA synthesis should start. So they act as a what? As a label or reference for the RNA to start, from which part it will be synthesized. These regions are important in the synthesis of proteins. They are very important in the manufacture and making proteins. After that, we need to get some information about mutations. As we studied, the DNA is very stable, but if the two parts cannot be repaired, it gonna be what gonna be happen? Something which is called mutations maybe happen at this part. Well, mutation can be also defined as a sudden change in the nature of the hereditary factors that control the sequence of DNA. Okay, that controlling a certain trait, I mean certain character, which leads to what may be desirable and may be undesirable character to change in these traits or this character in the living organism, whatever, as we said, desirable or undesirable. The mutation itself is considered true if it transferred through a different generations. I mean, if in each day we have a lot of mutations take place inside our body, okay, but they aren't transferred to the next generations, so they are just and they are gonna be repaired by the repairing DNA enzymes. So they aren't a true mutation, but the mutation becomes only true if it transferred to the next generation and so on. We have to distinguish and to differentiate between the mutation which results from the changing or alteration in the structure of the hereditary factor and the changes or alteration which is results from the environmental effect, the environmental impact itself. We have a great difference between the hereditary factor, the hereditary criteria, and the impact or effect of the environment, or from the accumulation and recombination of genes, or as we said it, the segregation of the genes. Most mutations lead to undesirable traits. Undesirable means that we don't need it yet. We don't need it much more, okay, as most of the deformation that happens in human, okay, like forked lips and whatever, crossing eyes, and a lot of mutations that we do need, a lot of the undesirable traits that we don't to see or to gain it. Mutations also in plants may lead to their sterility, okay, and that causes what? Deficiency in crop yield of these plants. There is no much more crop, that may causes the financial disaster. So in rare cases, I mean some rare cases, not much more, the mutation leads to some desirable changes. Okay, like the color of the eye, like the way of thinking, okay, and so on. But as we said, most of them, they are undesirable, like the autism in human, it's undesirable, and it's kind of what? Mutation. And man tries all the time to find a scientific ways to induce this mutation artificially. I mean the desirable mutation, okay? So, and definitely the most of the experiments are carried out via the plants and or by using the plants to get a desirable plant trait. For example, in American farmers discovered in his sheep, one of them has a short and curved legs. Okay, this is a mutation. But what is the desirable in this mutation? Let's discuss this nice example. He considered, this farmer, the American farmer, he considered this is a desirable trait because of what? As it cannot climb, the sheep couldn't climb over fences. It cannot climb over the wall, okay, and damage the cultivated plants. So he considered the curved legs as a kind of desirable trait. The farmer took care of breeding these mutant sheep, okay, as he describe it, until it gave rise to a new strain known as the Ancon strain. So he gave it this name as a kind of desirable trait for couldn't or it doesn't has any ability to jump over the fence and damage the crops. Other examples of desirable mutations, the men who has ability to induce crop plants in order to increase their yield, I mean their amount. We have different kinds of mutations, such as what? Mutations are divided into two main kinds. The first one, the genetic mutations, and the other one, which is what? Chromosomal mutations. So from its name, the genetic mutation or the gene mutations, they occurs or happen to the gene itself, while the chromosomal mutation, it occurs via the chromosomes or the changes that happen to the chromosomes. Genetic mutation or the gene mutations. These are due to what? Chemical changes in the gene's structure, also the arrangement of the nitrogenous bases, okay, that as we said it, adenine, thymine, cytosine, guanine. If the arrangement of the sequence in part of DNA, if it replaced or absolutely changed, it gonna or gives us what? The genetic or the gene mutation, okay? And also it maybe happens chemically. These changes finally leads to what? Production of a different enzymes, which would in turn develop a new trait, gives us another trait, okay? Any change also in the chemical structure of the gene is often accompanied by change from dominant to recessive state. Dominant, I mean the controllable one, the main one, okay, the most common one, to the recessive one or to the most retreated state, the most retreated trait. Although the opposites may occur in some rare cases, okay, I mean not the most common, no, but just the rare cases. And any change in the chemical structure, as we said, it necessarily to accompanies what? With the changing from dominant to recessive one. The chromosomal mutations here These mutations occur by how many ways? 2 ways. Occurred by what? 2 ways. They are what? Number 1, it may change in the number of chromosomes itself. Okay, we can miss a number of chromosomes. We have 4 or 6 chromosomes in each somatic cells, and this number, it may be increased or decreased. And this disorder, we call it in science, or we call it in biology, what? Change in the number of chromosomes. Yes, we call it the chromosomal mutation. This means loss or gain of 1 or more chromosomes during gamete production in the process of meiosis. Like what? We have some diseases or some disorders like Klinefelter, Turner, and both of them, they are kind of syndromes in man. Okay, each one of them has some of the characters and features, and they are actually they are very undesirable traits for human. Klinefelter and Turner syndromes. The cells contain also 1 or more extra chromosomes, as in the first case, or the cells may contain 1 chromosome less, as in the second syndrome, which is what? Turner syndrome. The number of chromosomes may be duplicated in a cell due to the non-separation of the chromatids after centromere division. At the moment of meiotic division, the moment of division of the centromere lengthwise, sometimes it doesn't happen, and that causes also chromosomal mutation. And failure of membrane formation between the 2 daughter cells. That means causing something which is called polyploidy. And this type, it's a very rare also and undesirable trait. This phenomenon of the polyploidy may take place in any living organism, okay? And most common, it happens in plants, where the large proportion of known plants have polyploidy. They are not haploid and diploid, no, but they are tetroid, hexaploid, okay, and also maybe octanoid, and in some rare cases they are sexteme. Okay, this is phenomenon which is called polyploidy. They are much more common in plants, and they are actually we consider it kind of, it's a very rare in animals. Where the chromosomes are duplicated during the gamete formation, what happened? This results in the production of individuals with the new characteristics, okay? And this is because each gene is represented by many copies. Each one of them has effect become more pronounced, become more clear, and appears in the individual. So the plant becomes taller, and consequently its organs become bigger, especially flowers and fruits. So if you observe a large flower or large stem and so on, something that's abnormal, you have to think in something that this is a mutation, but in some cases it may be desirable for the plant. We need to get a large fruit and a very tasty and a very delicious one. And nowadays there are many tetraploid crops. Tetraploids mean that they have a polyploid from type of 4N. And fruits such as cotton, wheat, apple, grapes, okay, pears and strawberries and so on, just to get what? To get the largest fruit, okay? So this is a mutation, but induced by human, and we call it an inducible one or induced mutation for a desirable trait.